So in the summer we had seen that there is an algal boom. So we will be looking at that algal boom. There is a favorable condition during the summer time for the coccolithophores. So they release or they are responsible for the algal booms. So once the algal boom happens, this actually has an impact on the local system. And then what happens is the death of coccolithophores occurs, which leads to sedimentation. That means they are actually settling down and they are going to the bottom of the sea. So from that they either contribute to sediment or the alkinone becomes part of the sedimentary record. So because the algae booms was the main source of alkinone production. So once they are dead, they directly go and because we had seen about the sediment analysis. So how do we extract alkinone from the sediments? That we had seen. Now we are seeing why, or how it is actually happening. So the algal boom, after the algal dies, they reach the sedimentation or they reach the bottom of the ocean, and from there they contribute to the sediment. Then they incorporate into sediment. They are found in other organic materials, and they are enhanced preservation in anoxic conditions, because their anoxic means the amount of dissolved oxygen is less. When the dissolved oxygen is less, what happens is many decaying materials, bacteria and other things don't survive at that time. So in other words, whatever is there will remain like that. They will not be degraded very quickly under anoxic conditions. So otherwise these alkinones become the part of sedimentary record. So that we will collect later on, maybe after few years or few decades. Then we can study that, okay, how much alkinones were present and all those things we can actually carry out the studies. So how the alkinones are basically formed? The source of organism is haftophyte algae. So they are also called as coccolithophores. They produce alkinones. When do they produce the alkinone? They produce during the algal booms. How does it actually occur? Because of the nutrient rich waters, because we are talking about the Arctic water systems. So the nutrients are there more in the Arctic water system, and these algae actually produce these alkinones. And then once the nutrient rich water is there, that upwelling temperature changes and nutrient flux are triggered by the formation of alkinones. So that is how more and more alkinones are basically produced. And if you look at the biochemical process, from the fatty acid precursors, there are certain enzymatic reactions that occur that produces alkinones. This basically depends on the environmental condition, temperature. So this is what we are correlating. When the temperature is right, then there will be more and more alkinones will be produced. So that is how we try to correlate temperature with the production of the alkinone, which is seen later on. And types and ratios of the alkinones are basically dependent on the environmental conditions, temperature and nutrient availability. So this functions as a proxy for our studies. And how the coccolithophores, algal booms and sedimentations are interrelated to each other. Basically, we can look at coccolithophores, alkinones and algal blooms in the Arctic region. So one is a biological relationship. So the coccolithophore basically rapidly proliferation leads to algal bloom, and if there is a hypoxia, that affects the other marine life also. Otherwise they survive, all the algals survive. Otherwise these algal blooms contribute to diversity of the species that are seen in the Arctic region. Similarly, coccolithophores are also involved in various roles in the ecosystem. They actually impact the food webs also. Otherwise, what are the different types of microalgae? They are basically the primary producers. They are part of the phytoplankton community. So they are the producers of food in the Arctic food web. And these coccolithophores also were food for zooplankton. Basically, the zooplankton consume these coccolithophores, and the zooplankton is being consumed by fish. They are consumed by bird, then they are consumed by marine mammals. That means this is the entire roadmap for the Arctic food web. So starting from the coccolithophore, zooplankton, fish, bird. From the coccolithophore, we go to the zooplankton, then fish, then bird, then marine mammals. The entire food web is covered here. And if you look at the chemical relationship of if you look at the chemical relationship of coccolithophore alkenones and alkyl blooms in the Arctic, the coccolithophores produces the alkenones. Due to their stability in sediments, they can be used as a biochemical marker. So that is what we have seen so far. The alkenone also is a concentration in the sediment cores talks about the environmental fact indicators that what existed in the past, that we can actually do a back calculation to find out. We could predict what could have happened during that time. Similarly, the coccolithophore contribute to carbon cycling, one in the photosynthesis, also they are involved in the carbon fixation by formation of carbonates in the seawater. Similarly, the coccolithophore also is used as, they use nitrates. The coccolithophore uses nitrates, phosphates and silicates for this affects the entire nutrient dynamics in the Arctic region. This also affects the growth of other phytoplanktons. Similarly, when the coccolithophore is decomposing, they can be used by other phytoplanktons also. So this is the overall role of coccolithophores in the entire Arctic ecosystem. So let us look at the relationship between U37 and SST. This is what we have been talking so far. Like the presence of alkenones can be correlated with sea surface temperature. So how they are related? The SST, that is the degree centigrade, is equal to A plus B into U37. So what are A and B? The A and B are basically coefficients. They can differ based on the specific ocean region being studied. So the local calibrations are often necessary for more accurate results. So we have to use a calibration curve. So how do you use the calibration curve? So here is the calibration curve. SST degree centigrade is equal to 47.1 into U37. That is the concentration of all the alkenones minus 2.6. And the environmental factors, how do they affect? The U37 index reflects not just the temperature but also other environmental conditions including nutrient availability and algal community composition. So this is what we have seen just now. What is the limitation of using this formula? The U37 is generally used as a good proxy, but it is only of several tools used for paleoclimate studies. That's what I said in the earlier also. This can be used as one additional tool and this is not the only tool. So that is the difference we have to keep in mind. And it estimates should be considered in conjunction with the other data sources for a comprehensive understanding of the past climate. So just based on U37 concentration or the alkenone concentration alone, we cannot predict what was the past temperature. So what are the implications in the paleo environmental studies? These are proxies for temperature, so they are extremely good. The relative abundance of the different alkenones can indicate past sea surface temperature. The U37 index, which compares the concentration of specific alkenone type, is commonly used for this purpose. We can also record the environmental change. We can also study this using a geological timeline. Basically, the presence and the composition of alkenone in sediment layers, not in one particular region, but in multiple layers, can provide insight into the timing and nature of past climatic events such as glacial interglacial cycles. So how do we study the Arctic water and the carbon cycle? We can use remote sensing, field measurements, hydrological models, carbon cycle models and climate data analysis. All these can be used to study the Arctic water and the carbon cycle. So what are the tools and models we generally use? One is the remote sensing tools, which is synthetic aperture radar with passive microwave sensors, or carbon monitoring satellites can be used. With respect to the hydrological models like SWOT, that is soil and water assessment tool, or variable infiltration capacity, can be used for measuring and studying this. Carbon cycle model include CASA, that is Carnegie Ames Stanford approach, and Joule's Joint UK Land Environment Simulator. Climate model include coupled model intercomparison phase, project phase 6 can be used. And data analysis tools include Python libraries, MATLAB and R for statistical analysis.